and as well as Prudence, for putting together this wonderful event. So also I want to, appreciate RIS for making me a speaker for this wonderful event. Since, as Yimin mentioned, right, there are many great researchers who have made great contributions to this particular field, protein engineering and directed evolution. What I'm doing here is just trying to navigate through those literatures, many of which actually dated back to decades ago, and try to figure out what has happened, and more importantly, what might happen in the near future, enabled by this amazing technology, so-called directed evolution. So let's begin with some important challenges facing the field of chemical and biological engineering. So I want you to think about these challenges, and through this lecture, at the end of this lecture, I want some of you, to be able to come up with certain solutions, or at least some ideas in terms of how to deal with these challenges. So the first challenge is widely known in the field of biofuel industry. Okay, people are looking for biocatalysts, particularly known as enzymes, that can convert biomass into small molecule sugars, which can be further utilized by living organisms to produce biodiesel in a very efficient manner. So one requirement is that these biocatalyst enzymes can survive, can work, function well at high temperature. In doing so, they can be adapted or integrated with the existing infrastructure used for chemical plants. A second challenge would be opposite. Can we develop a mild condition that will allow us to convert methane to methanol? Nature is able to do it. There are some microorganisms living in the bottom of the ocean that produce the enzyme known as methane monooxidase, a protein that can convert methane to methanol. But that enzyme is very complicated, very small, or very big, okay, and also unstable. The turnover rate is very slow. So can we find a way to design an enzyme that can do this, can do it fast, can do it under mild physiological conditions? So if we can do that, we will save a lot of energy. We can also make better use of natural gas for producing, gasoline or other electricity generation, for example. The third one is, can we enrich uranium from the ocean, from seawater? So this is known as one of the 7 chemical separations, ranked by nature. Let's say, it could change the world, right? Nuclear power is one of the relatively clean energy, okay, that comes with low carbon emission. But the problem is the current nuclear power industry heavily relies on the uranium source that's derived from land mining. The process can lead to pollution in the groundwater system, irreversibly. On the other hand, the ocean contains a vast amount of uranium, but the problem is that concentration of uranium in seawater is extremely low. There's no economic and efficient way to enrich them. So can we come up with a chemical process to address this problem that may be able to enable the humanity, last for another 8,000 years? What's the coming one? So we talk about the energy, right? The fourth challenge would be biomedical related. Can we develop stable protein drugs without complicated formulation? These drugs can survive in our blood serum, okay? That can reach, the disease site. So I just give you one simple example. This is arginase, bacterial arginase. Already showed exhibited the activity that can inhibit the growth of tumor by depleting arginine, one of the building blocks for protein synthesis. The problem is this bacterial arginase is very poor in terms of this reaction, okay, catalyzing this reaction. Also, they got quickly inactivated in the serum. Can we find a way to improve, optimize this enzyme to make sure they can survive and ensure their success inside human body? And then we can convert that into a useful therapeutic. Can we do that? The fifth one is more fundamental, okay? If you look into central dogma, DNA, RNA, protein, what do they share in common? They're all linear polymers. Okay, in terms of topology, they're simple. So the question I want to ask is, can we use cellular machinery to make nonlinear polymers? So if we can do that, we may be able to have access to new materials with properties that have never been explored before. So these are the 5 challenges I just presented here. So is there any simple yet universal algorithm that may help us address these challenges? Is there any universal one? So there is an old saying in China, right? In Chinese, so we call it 师法自然. Okay, this is according to Taoism. Okay, this is a philosopher, came up with this idea, thousands of years ago, right? We need to learn from nature if we are puzzled or facing difficulty in our lifetime. Okay. So we enjoy great ecological diversity now, which was made possible by Mother Nature. Okay. How did nature achieve this? So somehow, nature figured out a way to write using simple building blocks. Okay, like ATGC, to write the script of a very complicated script known as life. So, in which structure and a function at the molecular level can arise. Okay? So can we follow what nature has done and learn from nature? Repeat the process here. Can we write a script, a string of sequence that compose simple building blocks to derive function, derive structure and function from these combinations? This problem in the field of protein science and protein engineering is also known as protein folding problem. Can we deduce 3-dimensional structure or function from primary sequence of amino acids? Also known as design problem. Can we, if we have a function or certain structure in our mind, can we do the other way? Come up with a string of sequence that will lead to the structure. Just to use 20 different building blocks used by nature. Can we do that? It's not a simple question, right? Simple problem. Because imagine I just give you one example here. Think about this. Insulin, the smallest protein in our body, inside our body. It consists of 51 amino acids. So in order to design something that as useful as insulin, we have to, nature already achieved this. Nature is able to, search through this vast collection of combinations of amino acid sequences, which constitutes a space known as protein sequence space. This number is huge. Okay, 20 to 51. This is big number, much larger than the housing price here in Hong Kong combined. Okay, and also it turned out that the protein sequence space, if you notice that, it's very vast, empty. Now, some of you may like it because we live in Hong Kong. We're looking for space that's vast and empty. But for protein engineers, that's not the case. We're looking for something meaningful in this vast, empty space, which is full of nonsense. Okay, can we say, hold a stone of hope out of this mountain of despair, or can we find something meaningful out of this, of telos, purpose, whatever you call it, out of this vast space? And also, there's another terminology to describe the function. It's called fitness. You can define it. If we are interested in catalytic efficiency, we're just looking for, improved reaction rate. If we are interested in, say, thermal stability, we just look into can be called fitness. Okay, it can be any parameter or physical property you are interested in. Can we do that? How can we walk through this protein sequence and identify something that's useful? So what nature has done, nature used a tool known as evolution, right? They just randomize or change certain amino acid composing in certain size regions, and then they were able to walk through the protein sequence space, step by step. It's known as evolution. But what's the purpose of evolution? We know there's a rule in natural evolution called survival of the fittest. Okay? But in our daily life, when researchers conduct their experiments in the lab, we have a purpose. We want to inject a sense of meaning into directed evolution. Into evolution. This is called directed evolution. Okay, we want to inject a purpose for protein evolution in the laboratory. So, can we evolve protein molecules in the laboratory? This is not the idea actually come up with by Francis Arnold. Actually, one of the greatest chemists in the 20th century, Manfred Agent, who is in the field of fast reaction kinetics, okay, proposed this or envisioned the possibility of evolving protein molecules in test tube. But also he laid out the challenges we have to overcome in order to achieve this. One of the greatest challenges is that we have to be able to connect the genotype, which is carried by DNA sequence, with the phenotype that is carried by protein molecules in a surest way. Okay, we have to connect them because they are carried by different molecules. Can we reconcile this distinction in the process of directed evolution? Because we are changing DNA sequence, and in the end, we are looking into the protein function. Okay, can we reconcile that conflict? And also, can we construct a machine that will enable us to quickly look into the function of these protein molecules in a high throughput manner? We have to overcome these two. So, one decade later, it is Francis Arnold and her student who were able to demonstrate we can achieve protein-directed evolution in the test tube. Okay. What they did is just they introduced random mutagenesis into a protease, which is known as certilicin. Okay? And then, after multiple rounds of random mutagenesis, they did an aerochrome PCR. They can play with the condition. They reduce some amount of metal ions, and then they change the stoichiometry between DNA building blocks A, T, G, C. They can play with the stoichiometry. They were able to make the amplification process error-prone. Okay? By doing this, do it repetitively, they were able to evolve a catalyst, an enzyme that can work in the presence of huge amount of organic solvent. So this becomes one of the first enzymes that can survive in 70% of organic solvent, known as formaldehyde. Okay, this is significant. And through this work, Francis' group was able to master the flowchart workflow for directed evolution. Basically, she summarized 4 important steps that's essential for directed evolution. First, you need to be able to identify a starting point, a parent protein that can be used for directed evolution. One thing, you have to find a very low activity of the enzyme. You cannot evolve something from nothing. Okay, you first have to find an enzyme that carries some low-level activity, and then you subject that to randomization. You introduce mutations to work through the protein sequence space step by step by introducing limited randomization. It's called an error-prone PCR. And then you have to come up with this is very important, okay? This is the essential for the success of directed evolution. You have to be able to find a way to quickly assay all these genetic variants in a high throughput manner. Okay? They did it in 96 well plate format in early days. Okay, that's the most commonly used one. And then you find those, the fittest, the hits, we call it the hits, okay, that should improve tolerance toward organic solvent in that case. And then you just subject it to another round of directed evolution until the behavior of the protein or the property of the protein can satisfy our need. Okay. So what's the problem? Think about this. If we just do random mutagenesis, what's the problem? At a certain stage, after certain round of directed evolution, you may end up with multiple hits or candidates that showed comparable efficacy or reaction efficiency or tolerance toward organic solvent. How can we move further? If we have multiple, like say you have 5 candidates that showed similar improvement, how can we move further? So there was another guy, William Stemmer. Just 1 year after Francis published their work in PNAs, William Stemmer came up with this solution called DNA shuffling. They were able to combine these genetic variants in test tube to do in vitro recombination of these genetic variants. They do hybridization in vitro. What they did is they can mix these parent genes and then digest it with DNA, chop them into fragments. And then they were able to find a way to repeat them together to form chimeric genes. Then, in doing so, they were able to create a library that contains, genetic variants that are derived from those parent genes. And in doing so, they were able to propagate these beneficial mutations from the previous rounds of directed evolution. They were able to accumulate those improvements in the following genetic descendants. That's what they did. And in doing so, combining random mutagenesis and DNA shuffling process, they were able to facilitate the process of directed evolution. So the message here is that if you can do both random mutagenesis and that DNA shuffling, in principle, we can evolve any enzyme or protein molecules in our lab. Just need these two great tools. And of course, because of this groundbreaking contributions, both Francis Arnold and William Stemmer were awarded with this Draper Prize in Engineering. This is also known as a Nobel Prize in Engineering, because there's no real Nobel Prize for engineering, so this is the one that is as important as Nobel Prize. Sadly, William Stemmer passed away just two years ago because of brain tumor. Otherwise, I'm pretty sure the Nobel Prize this year would go to Francis Arnold and William Stemmer. That's the sad story, but life is complicated, but science is always fun in this process. So, following this DNA shuffling, came up with by William Stemmer, Huimin Zhao, one of the earliest Francis Arnold students, further improved this in vitro recombination process. Basically, they were able to combine error-prone PCR and this in vitro combination process in one PCR reaction. It's just a change of the protocol for PCR. You can do both in one step. That's what Huimin did. It's called a step-based in vitro recombination. And actually, Huimin will come, I think, mid-December to give a talk about biosystems design enabled by directed evolution. His lab was able to apply directed evolution principles into engineering of complicated biological systems, not only protein molecules, but all the other living systems. So, because once we have DNA shuffling and error-prone PCR, we can do a lot of things. That's indeed what the past 2 decades have witnessed. So there are several important applications. Some of you may be familiar with. This is one Nobel Prize winning technology. Basically, Osama discovered this green fluorescent protein. It's genetically encoded in jellyfish in early 1960s. And then 3 decades later, another bioengineer, Roger Chen, took over. They applied the principles of directed evolution to quickly diversify the toolbox of genetically encoded fluorescent molecules. They started from 2 parent proteins. One is called a green fluorescent protein, the other is called red fluorescent proteins. They were able to quickly expand the collection of these genetically encoded tools. And these tools are nowadays widely available, widely used in biology labs, okay? And this is just one example. And another example would be, if you still remember, one of the challenges would be, how can we design proteins that can convert small molecule alkyne into alcohol? If we can do that, we can revolutionize the oil industry, okay? How can we do that? So, in nature, there are a variety of enzymes known as cytochrome P450. This is the enzyme can convert long aliphatic acid into, do oxidation to install oxygen molecule into these long aliphatic chains. So, build upon this natural enzyme, through multiple rounds of directed evolution, Francis Arnold's lab was able to convert this enzyme into this so-called ethane or propane monooxygenase. So this enzyme can oxidize those small alkyne into small molecule alcohols. So this is what they did. It's decades of effort. Okay, they were able to oxidize, to generate ethanol by oxidizing ethane. What else can we do? So Michael Shapiro, in collaboration with Francis Arnold's group, come up with even crazy idea, okay? This P450 can not only be used as a catalyst for certain organic reaction, it can also be used to detect, as a sensor to detect important small molecules inside of the body. What he did is just introduce several mutations through random mutagenesis and in vitro recombination. They were able to convert this enzyme into a dopamine sensor. It's an MR contrast agent. How does that work? So basically, without the substrate, dopamine, okay, this enzyme, the active site is occupied by water molecules. Because this water is close to the cofactor, it's a heme center, contains a ferric iron 3. And then it generates MRI signal. So once you have dopamine level increased, so the dopamine will kick out water molecules, and then MR signal will be gone. That's the principle. How we can take advantage of these engineered proteins to image the distribution of dopamine in the nerve system, okay, inside of the body. So what else? Arnold's group also demonstrated that just started from a bacterial or some simple proteins. It's known as cellulose, derived from the fungi or some bacterial cells. This is an enzyme that can convert biomass like cellulose into small molecule sugars, okay? But the problem is the natural one is very slow in doing this type of reaction. As some of you have some chemistry background, okay? How can we speed up the reaction? The most obvious way would be, can we raise the temperature, speed up, accelerate the reaction? If you increase temperature by 10 degrees, the chance is good you are going to increase the reaction rate by 2 to 3-fold. That's the thumb rule, okay, widely known by junior chemistry students. But the problem is, many protein molecules or enzymes can work under mild physiological conditions. That's a narrow window from 4 degrees to 37 Celsius. So Arnold's group was able to evolve an enzyme, through multiple rounds of directed evolution to improve its stability, thermal stability. So in the end, these work, okay? At the end of the day, these enzymes can survive and even work well at 90 degrees. Okay, that's what they can do with directed evolution to dramatically improve the thermal stability of protein molecules. So I think, after 2 decades, right, the lab should be, that's the moment when I joined the lab, okay, the lab should be complacent. So people, the idea has been circulating around, people flirting with the idea that Francis may win Nobel Prize, okay, in near future. It sounds to many junior students, okay, we don't have to do anything, we just sit and wait, okay, to harvest the big prize, okay, that's all we need to do. And, actually, that's the case, right? In 2011, Francis got this awarded the National Medal of Technology. Okay, this is it, the photo of him, Barack Obama and Frances Arnold. So Draper Prize, that's another big award for her lifetime achievement in the field of engineering. So it seems we indeed had a great time back then. I joined the lab in 2012. This is a golden era, and for Arnold lab, and I have to correct Yimin because Frances at least is always nice to me, and I never see her be angry or be show any bad temper. Okay, that's not the case. She's always supportive. When I ask for recommendation later, she said, okay, just let me know where you want to apply. And also she keeps sending me email, these wherever there's a job opening, she just forward me the email and let me know the most updated information about the job market. Also another thing, I still remember in a sweltering summer afternoon in California, okay, she would just walk me across the campus to visit a very junior assistant professor. Another lady, as Viviana Gradinello, another neurobiologist on campus at Caltech, walked me half of the campus to visit that young lady to talk about the potential collaboration. Okay? So, not the other way. She didn't ask that young lady to come over and we talk, okay? This is opposite. So, she's very down to earth in this sense. Also, she always enthusiastic about science, okay? We didn't talk about, in group meeting or whatever social gathering. She didn't talk about things randomly, okay? She just sometimes ask about your personal life, whether you are okay with the lab environment or culture, or whether you can do something that's more, that's different, okay? You can distinguish yourself from others in the lab, okay? That's another mindset which I can cultivate in Frances by working with Frances Arnold. So we had a good time, This is the first day I joined the lab. We had a big party. The party is not for me, but I happened to, to enjoy that party during my first day at Caltech. And it's nice. And now the lab size is increased dramatically, okay? Trust me. You can go online and find out the most updated photo. For the group, we did a lot of hiking, and this is the cabin owned by Frances Arnold in this valley, okay, near Arcadia, another small city near Pasadena, okay? This is the cabin. I'm not sure whether it still exists there because of the fire, okay? We had a good time, but I think it turned out that after 2013, it appears to me science or research in Arnold lab turned out to be more exciting than, at least to me, okay, it's more exciting than when I navigated those older literatures. Because with directed evolution and with the accumulation of bigger library of these enzyme variants, P450 for example, we were able to really dig into these libraries and identify new chemical reactions that doesn't exist in nature, but can be catalyzed by those genetic variants, okay? So another thing I want to remind you, because this is something you have to keep in mind when we discuss about further developments about the directed evolution, okay? So if you want to evolve a new chemical reactivity, you have to keep in mind, when you do directed evolution, you have to begin with some enzyme that is promiscuous, okay? We said that the natural evolution doesn't have a purpose, doesn't have a telos, okay? They just sit there waiting for us to discover those promiscuous activity. Although, we said that this P450 enzyme is good at oxidation to convert aliphatic alkyne chain into alcohol, right? But it depends if you just artificially mix the enzyme with some other bizarre substrates. Some new activity may arise, okay? You just wait for us to discover those new reactivities. As long as we can get some low minimal level reactivity, then we can